everyone, welcome to lesson 1.1 and the first lesson covered in grade 11 biology. In this lesson, we're going to be going over an introduction to cells, as well as a brief review and overview of the grade 10 biology curriculum. However, we're going to be going more in depth and more in detail in concepts and knowledge regarding the biology and physiology behind cells as a whole. Now, before we begin the lesson, just a quick disclaimer for this unit and future units is to not treat these video resources as an exhaustive resource to use for tests and evaluations. Please remember that your teacher is the one that prepares these tests and evaluations for you, and so your teacher is who dictates what knowledge and concepts you need to be familiar with from the curriculum. So please keep that in mind in future lessons and videos. So without further ado, let's get into the lesson. Cells. So cells are the fundamental building blocks of life, and they make up all living organisms, from animals to plants to bacteria. The smallest organisms are unicellular, and they are composed of only one cell. The largest organisms are multicellular, and they are composed of multiple cells. Now, cells can vary in size and shape, but they all share common features that are universal throughout all cells. So all cells are surrounded by a membrane that can protect the cell from foreign substances and allow other substances to enter it. All cells also contain genetic material, which is the storage of instructions that carries out all of a cell's duties. Cell activities are chemical reactions that are catalyzed or caused by enzymes produced inside of the cell's cytoplasm. All cells also have their own energy release system that powers all of the cell's activities. Alright, so moving on to a brief history of cells. So we can see through this timeline that various advancements have happened throughout the history of cells and cell research. Starting in 1655, when scientist Robert Hooke observed what we now know as cells of a cork tree through a primitive early prototype of the microscope. This is when the term cell was first used and coined. To 1674, when the observation of the first living cell was ever discovered through the discovery of protozoa. To 1838, when the cell theory was formulated, which provided us with an operational definition of life. All the way till 1997, when Dolly the sheep was the first ever organisms to be cloned. Alright, so moving on to the actual cell theory. So the cell theory has three main essential ideas that formulates the entire theory as a whole. So the first point and idea of the cell theory is that all living organisms are made up of cells. The second point and idea is that cells are the basic unit of life. And the third point is that cells can only come or be created from pre-existing cells. So cells cannot spontaneously generate, and they must be created and can only be created from the division of pre-existing cells. All right, so moving on to the 7 functions of life. So in biology, we have certain criteria that we must apply in order to classify whether or not something is living or non-living. And this criteria is known as the 7 functions of life. So metabolism is the first function, and it is the chemical reactions that occur inside the cell of a living organism. It's also known as cell respiration. Then we have reproduction, in which all living things and organisms must reproduce either through sexual means or through asexual means. Homeostasis is the next function, and this is the maintenance and regulation of internal cell conditions in an organism. Excretion is the fourth function, and this is the excretion or expelling of waste by an organism. Response is the fifth function, and this is the ability of an organism to react to changes and stimuli in the environment. Growth is the sixth function, and this is when living things and organisms can move around physically and change in size and shape, usually over time. And the seventh function is nutrition. So all living organisms must obtain food through one method or another to provide energy for themselves and materials that are needed for growth. And the 7 functions of life can be remembered through this abbreviation, Mr. HERGN. All right, and just a quick note on multicellular organisms. So in multicellular organisms, identical cells in a group will organize themselves and interact with each other to form a living organism with distinctive, unique overall properties. And because of this, multicellular organisms can show emergent properties, which are properties that an entity gains when it becomes a part of a larger system overall. This can be seen here in this image. So we have single cells right here. And these cells group together and interact with each other in such a way that tissues are formed, or epithelium tissue, so skin tissue, is formed. Then these tissues will interact with each other to form organs, such as the stomach. And then these organs will interact with each other to form organ systems of a living organism, such as the digestive system. And then these organ systems will all interact together to create a full living organism that has emergent properties. All right, so now we're going to be moving on to cell differentiation in multicellular organisms. So, as you know from previous biology knowledge, different cells will perform different functions for an organism. So, for example, red blood cells will carry oxygen, and retinal cells will absorb light and transmit the impulse to the brain in order to interpret colors and images. So, cell differentiation is a process in which cells will become specialized to carry out their role more efficiently than if they had many different roles combined. And in humans, we have 220 distinct, highly specialized cell types to perform different functions for our bodies. So, gene expression and its correlation with cell differentiation. So, when a gene is being used in a cell, we can say that a gene is being expressed by the cell. And this essentially means, in more basic terms, that the gene is switched on and the information is used to make a protein in the cell to assist in creating a function for the cell. So when cells develop, certain genes will need to be switched on and expressed, and others will need to be switched off, so they are not expressed. And the control of gene expression is the key to development of cells. We can see that right here in this diagram. So we have a single cell right here. This cell will undergo mitosis, or as you know, cell division. It will split off into two identical daughter cells. This daughter cells will express a neuron-specific gene, and so it will become a neuron. This cell will express an epithelial-specific gene, and so it will become an epithelial cell. And that's essentially how gene expression works in cells. Right, moving on to stem cells. So stem cells are undifferentiated cells that can have the potential to differentiate into various types of body cells and replicate and divide indefinitely to produce more of the same cell type for multicellular organisms. So as you can see, this can be very beneficial to researchers in creating and developing new treatments to treat certain conditions and diseases through the usage of stem cells. And we're going to get into that in a little bit, but for now we're just going to focus on more so how stem cells can be created and extracted. So as you know, when a sperm fertilizes an egg, a zygote is produced. And this zygote will divide to produce an embryo. And cells at the 2, 4, 8, and 16 stage in the embryo are pluripotent. And this means that they can differentiate along different pathways into any cell type in the body. And that's essentially how stem cells are created naturally. And they are essentially called stem cells and referred to them as stem cells because almost all adult tissues stem from them. Stem cells usage in research. So, stem cells have various useful properties to them in that they can divide continuously and replicate continuously, and this can be very useful for the growth of tissues or the replacement of cells that have been lost or damaged due to disease or injury. And stem cells also have the capacity to differentiate into specialized cell types in the body. Therapeutic uses of embryonic stem cells. So, embryonic stem cells are very useful to researchers and medical practitioners alike, as they can produce tissue for burn patients. They can potentially heal diseases where a particular cell type is lost or malfunctioning, and they can also potentially grow replacement organs as well if necessary. So, differentiation process. So, the early stages of embryonic stem cells are most versatile. As the embryo develops, the cells commit to a pathway of differentiation and then eventually to a specific cell type. So once a cell is committed to a specific cell type, the cell can divide, but all of the cells will differentiate in the same way and are no longer stem cells. All right, so moving on to adult stem cells. So adult stem cells are essentially when small numbers of cells remain as stem cells in the human body as it matures and enters adulthood. And these stem cells are present in many adult tissues, including bone marrow and skin. And they give some human tissues considerable powers of regeneration and repair. And they can also be found in other tissues in the human body, such as the brain, kidney, and heart, but they only allow limited repair in these organs and tissues. And just to put into perspective the therapeutic usage and treatments that stem cells can allocate, Bone marrow transplants can be used to cure leukemia as a result of stem cells. So, through the process, a needle is inserted into the donor to remove the fluid from the bone marrow. And then stem cells are extracted from this fluid and stored by freezing. Stem cells from the patient are then destroyed by chemotherapy, and thus the donor cells are returned to the patient's bone marrow to produce new healthy blood cells. And here's just a quick diagram that really puts into perspective the power and ability of differentiation of cultured pluripotent stem cells in terms of that can differentiate into a multitude of cell pathways, including bone marrow, which can be used to cure leukemia, nerve cells, which can be used to cure Parkinson's disease and paralysis, retinal cells, which can be used to treat blindness, myocardium, which can be used to treat heart disease, isolate cells, which can be used to treat diabetes, and skin cells, which can be used to treat burn victims. Uses of stem cells. So, therapeutic uses of stem cells include in vitro processes, in which embryos can be created by fertilizing an egg cells with sperm cells, and allowing the zygote to develop before pre-implantation and development. So that's one way of extracting stem cells, and another way is through obtaining them from the tissues that some adult stem cells can exist in. Now, moving on to the ethics of stem cells. So, most objections regarding the use of stem cells are centered around the use of embryonic stem cells, as current techniques usually involve the death of the embryo when the stem cells are extracted. And this really poses the question debate of when human life begins. So, some individuals believe that human life begins when the sperm fertilizes the egg, and some state that when there's a heartbeat or bone tissue or activity and development, that's when human life begins. And these stages will usually take place weeks after development. And others believe that when the embryo has developed into a fetus and is capable of surviving outside of the uterus, that is when life truly begins. So it's really centered around the debate of when human life begins. Some also argue that if embryos are created by in vitro fertilization in order to obtain cells, no human that would otherwise have lived has been denied its chance of living. And so this really poses onto the philosophical debate in question regarding that it's unethical and immoral to create human life only to obtain stem cells. And in vitro fertilization also involves hormone treatment of women as well as an invasive procedure to remove the egg. And this leads into debate that if women are paid for supplying eggs, this could lead to the exploitation of vulnerable groups in certain demographics. So that marks the end of this lesson for lesson 1 in unit 1. Thank you for listening, and I'll see you next time.